Okay, so welcome back to this next video in which we are discussing rheumatoid arthritis and anti-rheumatoid drugs. Okay, so we're in the process of discussing the adaptive immune response so that we can understand the autoimmune response that occurs in rheumatoid arthritis and underlies the disease. Okay, right. We have discussed so far about how the adaptive immune response is this response in which you activate B cells and T cells which are specifically targeted against an epitope of a certain antigen on the surface of your pathogen. Okay, so we've discussed that what it begins with is a antigen presenting cell, an APC, the principal example of which is a dendritic cell, which phagocytoses the pathogen, breaks it down, and then presents the fragment of your antigen, and we're looking at purple antigen, that corresponds to this specific epitope on its surface attached to an MHC class II molecule. Okay, now, in reality, of course, our dendritic cell is going to process the entire pathogen down here, and it's going to have loads of MHC class II molecules on its surface with loads of different fragments from all of those different antigens. However, to keep this simple, we're just thinking about this single epitope of purple antigen, okay, and that fragment corresponding to that epitope of purple antigen is the fragment that's presented on the MHC class II molecule. What's then going to happen is our professional antigen presenting cell is now going to travel through the lymphatics to a lymph node where it's going to meet CD4 positive T cells. Okay, now basically, what it now needs to do is find a CD4 positive T cell that has a specific T cell receptor which can bind to that peptide fragment that is presented on the MHC class II molecule. Okay, now in reality, of course, remember the dendritic cell will be covered in MHC class II molecules with loads of different peptide fragments on their surfaces, okay? So it will actually be looking for T cells that can bind to any of these, which is why this process can actually occur in a physiological time limit, okay? But for simplicity purposes, we've discussed that we're just considering this single peptide fragment corresponding to that single epitope of purple antigen, okay? So let's imagine our antigen presenting cell has found a CD4 positive T cell with a T cell receptor on its surface which can bind to that peptide fragment bound to that MHC class II molecule. Okay, then what will happen is the CD4 will bind to the MHC class II molecule, and this together will give signal 1 to this CD4 positive T cell. Now, signal 1, however, is not enough to actually activate the CD4 positive T cell. To activate it, you also need signal 2, okay? And signal 2 comes from the antigen-presenting cell presenting co-stimulatory molecules on its surface. Now, this is a checkpoint, basically, because only antigen-presenting cells which have come from a region that is actually infected with a pathogen will have co-stimulatory molecules on their surface, because they are put on in response to the activation of pattern recognition receptors on the antigen presenting cell by pathogen associated molecular patterns. Okay, so the principal examples then of co-stimulatory molecules are B7 molecules, such as B7.1 and B7.2, which are also called CD80 and CD86 respectively. These molecules will bind to CD28 molecules on the surface of our T cell and cause signal 2. Okay, signal 1 and signal 2 together will then cause a change in gene expression within our CD4 positive T cell, and it will result in the production of the interleukin 2 molecules and the interleukin 2 receptor alpha molecules. Interleukin 2 receptor alpha joins interleukin 2 receptor beta and gamma, which are constitutively expressed by the CD4 positive T cell to create functional interleukin 2 receptors. Okay, and then the interleukin 2 that you've secreted then binds to the interleukin 2 receptors on the same CD4 positive T cell, and then that delivers signal 3, and then this CD4 positive T cell with a T cell receptor that is specific for that epitope of purple antigen is now going to start proliferating. Okay? So it proliferates and proliferates and proliferates, and then these cells can start differentiating into T helper cells. And the main three types of T helper cells are T helper 1 cells, T helper 2 cells, and T helper 17 cells. Okay, in rheumatoid arthritis, the two main ones that they start differentiating into are T helper 1 cells and T helper 17 cells. Now, T helper cells are capable of helping B cells in the activation process, and all three types of T helper cells are all capable of activating B cells or assisting in the activation of B cells. Okay, and the reason that they're all capable of assisting in the activation of B cells is that they all have this CD40 ligand molecule on their surface. Okay, and this is going to be really important for helping in the activation of B cells. Okay, so let's now talk about B cell activation then. Okay, so B cells, or B lymphocytes to give them their full name, are also found mainly in lymph nodes and spleen. Okay, and B cells, like T cells, also are specific to a certain epitope of an antigen. Okay, so let me draw my B cell here. So B cells produce molecules called antibodies, and they're the only cells in the body which can produce antibodies, along with obviously plasma cells, but we'll consider plasma cells as being part of B cells. Okay, and the antibody molecule that a specific B cell produces is unique. Okay, so each of the different B cells sitting in the lymph node will produce a different antibody molecule. Okay, so now, as promised a long time ago, we'll now discuss the structure of antibodies in a bit more detail. So I'll draw a big picture of an antibody here. So antibody molecules are made out of 4 proteins bound together. Okay, 2 heavy chain proteins, which are these proteins that I'm now colouring in red here. Okay, and I should also say that antibodies have another name. Okay, antibodies are also called immunoglobulin molecules. Okay, so an antibody and an immunoglobulin are the same thing basically. Immunoglobulin is just a fancier word. An immunoglobulin is usually abbreviated down to IG, and an antibody is usually abbreviated down to AB. Okay, right, so these proteins in red are known as the immunoglobulin IG heavy chains, okay, and you've got 2 of those bound together in the middle here, okay, and then attached to the 2 heavy chains you've got 2 smaller proteins, okay, which I'll colour in blue here, which are then called the immunoglobulin light chains. Okay, so these are the immunoglobulin light chains. So all B cells are capable of producing antibodies, these molecules created out of 2 immunoglobulin heavy chains and 2 immunoglobulin light chains. And the antibodies produced by different B cell clones are going to be very different. Now, which portion of the antibody is it that varies? Okay, well, it's the portion that actually binds the antigen, and these are the portions of the antibody molecule which binds the antigen. Okay, so the tips of the heavy chain and the light chains. Okay, so these 2 regions, which are identical in the antibody molecule, so this region is identical to this region. Okay, they're just the mirror images of one another. These are known as the variable regions of the antibody molecule because amongst different B cell clones, they're going to be different basically. So different B cells are going to produce different antibodies, and the bit that varies is this variable region. Now the variable region has another name. Okay, it's also known as the FAB, standing for antigen binding fragment basically. Okay, and I will highlight this antigen binding fragment in orange here. Okay, so we'll often refer to it as the FAB from now on. Okay, then the rest of the antibody molecule, okay, so this bottom portion of the heavy chain and the bottom portion of the light chains, this is the constant region, okay, and the constant region is often abbreviated down to the fragment crystallizable because if you take this portion of the antibody molecules, it's capable of crystallizing, but if you like, you can think of this as fragment constant. In reality, the reason it was named that was for fragment crystallizable rather than constant. Okay, so we'll highlight this in green. Right, okay, however, I want to talk about one more little thing, which is that this constant region these B cells can produce antibody molecules with different constant regions. Okay, so let me just explain what I mean by this. So we've discussed how the variable region is different for different B cells. So if you go to different B cells, they're all going to produce antibody molecules with different variable regions, and these variable regions will be able to bind to different epitopes of different antigens. So if we've got antigen purple here, and remember we were studying a specific epitope of antigen purple, basically there will be B cells which produce antibodies which are capable of binding to that specific epitope of antigen purple that we are interested in. Okay. However, every single B cell is capable of also choosing different types of constant regions to use. Okay, so once you've got a B cell and it produces antibodies with a certain variable region that's capable of binding to a certain epitope of an antigen, it can then actually produce different antibody molecules that all have the same variable region but have different constant regions, and this goes for all B cells. Okay, so by varying the constant region, mainly the constant region of the heavy chain here, you can produce different antibody molecules which have different functions. Okay, and these different types of antibody molecules are called IgG molecules, IgA molecules, IgM molecules, IgD molecules, and IgE molecules. Whoops, I've done those in the wrong order, so never mind. So IgG, IgA, IgM, IgE, and IgD. So all B cells are capable of producing these different types of immunoglobulin molecules, basically. Okay, and all that varies here is which type of constant region you use here. Okay, so all of these different types of antibody molecules will have the same variable region as long as they're being produced by the same B cell. Okay, but it can choose to use different constant regions, and those create different antibody molecules with the same variable region. So all of these different types of antibody molecules will bind to the same antigen, but then the effect of the antibody molecule is different. Okay, so to give some examples here, IgG and IgM are absolutely fantastic at activating complement molecules. Okay, so basically these types of antibody molecules where you have this sort of a constant region, an IgG or an IgM constant region, these can bind to the antigens on the surface of pathogens, and then they can activate the complement cascade on the surface of the pathogen. So just to draw a little picture here, here is our pathogen, here is purple antigen, let's say. If we produce antibody molecules against purple antigen, that specific epitope of purple antigen that we've been very interested in all along, and these antibody molecules are either IgG or IgM antibody molecules, which refers to the specific type of constant region you're using here, then this constant region is then capable of activating complement proteins. Okay, whereas some of the other constant regions corresponding to these other types of immunoglobulin molecules, they are not capable of activating the complement proteins nearly as well. Okay, so that's what I mean by these different types of antibody molecules having different functions. Okay, right, so now we've talked about the fact that different B cells have different antibodies that they're capable of producing which have these different variable regions. We've also talked about the fact that a single B cell is capable of producing antibody molecules with different constant regions, and these different types of antibody molecules. Now what we want to discuss is going back to purple antigen. We want to launch a B cell response to that specific epitope of purple antigen. How are we going to do it? Okay, well, basically what's going to happen is B cells have receptors on their surface which are a special form of modified antibody molecules. Okay, so let me draw my B cell again here. So this is going to represent my B cell. So B cells put antibody molecules on their surface that are of the IgM or IgD type. Okay, and these special antibody molecules have been modified slightly, okay, to give them a little bit of an extension down here, okay, which is the portion that's going to implant into the cell membrane, basically. Okay, so B cells produce IgM and IgD molecules that have been modified, so that they're not soluble anymore, and instead they're going to be sitting on the cell membrane here. Okay, and they are going to have this special variable region that is specific to this B cell. Okay, and this receptor here is known as the B cell receptor, or for short it's called the BCR. Okay, so this just stands for B cell receptor, like TCR stood for T cell receptor. Okay, right, so now we need to activate B cells then that have B cell receptors that are capable of binding to that specific epitope of purple antigen. Okay, so how is this going to work? Well, going back to our picture of the lymph node, the B cells are sitting in the lymph node here, okay, and they're monitoring all of the lymph that is flowing through the lymph node. Okay, now basically the idea is that if the pathogen is present in our peripheral fluid, okay, what's going to end up happening is some of these purple antigens are going to end up breaking away from the pathogen, okay, and they're going to end up coming in the lymphatic fluid to the lymph node. So basically, some of these purple antigens that are on the surface of the pathogen are going to end up going into the extracellular fluid. They're then going to end up draining in the lymph fluid, okay, and therefore draining to the lymph node. And they're then going to be in contact with our population of B cells, okay? And now all we need to happen is we need that certain epitope of purple antigen to bind to the variable region of our B cell receptor on our B cells that produce antibodies specific for that epitope of purple antigen. Okay, so what's now going to happen then is, because the lymph is percolating amongst the lymphocytes in the lymph node, then eventually this purple antigen is going to meet a B cell with a B cell receptor which has that special variable region which can bind to this special epitope of purple antigen. Okay, so let's have this here. Okay, so here now comes purple antigen, and that special epitope of purple antigen that we are studying the adaptive immune response to is now going to have found a B cell that produces antibodies that are specific to that epitope. And we know that that's the case because the B cell receptor is just a special type of this antibody molecule that this B cell is capable of producing. Okay, right. Now that alone is not enough to activate the B cell. Okay, the binding of its antigen molecule to it is not enough to activate the B cell. It now needs what's called T cell help. It needs help from helper T cells, T helper 1, T helper 2, or T helper 17 cells. Okay, so what's now going to happen in this B cell is it's going to internalize the entire B cell receptor with the purple antigen molecule bound. Okay, so what it's going to do is to draw this, it's going to make a little vesicle inside which will contain, am I going to even try and draw this? I will try. Okay, it will contain the B cell receptor shown there, okay, with the antigen molecule bound to it, okay? So I'll just draw a bigger picture of that because I don't think that's going to be easily visible on the camera. So this is this vesicle zoomed in on, basically. Here now is our B cell receptor, okay, which is this special IgM or IgD molecule that has the membrane binding portion attached onto it, okay? And now you've brought in this special purple antigen into this vesicle. And now what the B cell is going to do is it's going to break down the contents of this vesicle. It's going to break down purple antigen here, and it's going to express, or rather put, fragments of purple antigen on its surface bound to MHC class II molecules. So B cells also have MHC class II molecules. Okay, so here is the MHC class II molecule here, and basically now in this MHC class II molecule you're going to have the little fragment corresponding to that specific epitope of purple antigen that we have been studying the adaptive immune response against. That's going to be put on at least one of these MHC class II molecules. Of course, in reality, the B cell is going to end up with loads of MHC class II molecules on its surface, and it's going to have loads of different fragments on those MHC class II molecules. Okay, but we're only interested in this specific peptide fragment that corresponds to that epitope that we've been studying the adaptive immune response against. Okay, so here it is. There's that peptide fragment. Okay, now on an MHC class 2 molecule. And now you can probably guess what's going to happen now. All it needs to do now is find one of these T helper cells which has a T cell receptor for that specific peptide fragment of purple antigen, okay? And then the CD40 ligand molecule can then activate the B cell. So let me now show this. Okay, so here, let's say this is one of our T helper cells. So it's either a T helper 1 cell, or a T helper 2 cell, or a T helper 17 cell. Okay, so it has on its surface then this T cell receptor, which is specific for that purple antigen fragment here. Okay, and of course what will also bind is the CD4 molecule that's on the surface of the T helper cell still. Okay, so it's still a CD4 positive T cell. Okay, so here's our T cell receptor shown here in red, and here's our CD4 molecule shown here in blue. Okay, right, so that's one of the things that then causes the activation of the B cell, but one of the really important things that causes the activation of the B cell is that this T helper cell, okay, will have on its surface the CD40 ligand protein here in orange, okay. So this is the CD40 ligand protein that we talked about was on the surface of the T helper cells, and this is going to bind to a receptor on the surface of the B cells called CD40. Okay, so I colour in the CD40 in purple here. And that then, together with the activation of the MHC class 2, which you get when the T cell receptor and the CD4 bind to the MHC class 2, is then going to activate the B cell. Okay, so this is what we mean by T cell help, basically. Okay, the activation of B cells is said to require T cell help. It's said to be T cell dependent. Now, the activation of B cells is a little bit more complicated than this, but for our purposes, we are now going to simplify this down to the B cell is now going to divide and divide and divide and divide and produce a whole population of B cells, basically. As I said, it is a little bit more complicated than that. Actually, what happens is a process of affinity maturation of the B cell receptor, okay, but we're not going to go into that. We're going to keep it nice and simple. Okay, so what happens then, effectively, is once the B cell has received this activatory signal from our T helper cell down here, it's now going to divide and divide and divide, and therefore you're going to get a whole population of B cells being produced. Okay, so here is this entire population of B cells, so we'll call this the B cell clone, because all of these B cells now are going to have the same B cell receptor. They're all going to produce antibodies of the same type, all going to produce antibodies that can bind to that specific epitope of purple antigen. Okay, and now what can happen is these B cells can differentiate into plasma cells, okay, which are those funny shaped cells in the oval shape like so, with their nucleus pushed to one side so that they can have a large antibody production factory here. And these are now going to start producing soluble antibody molecules, okay? And they can produce soluble antibody molecules of different types, basically. The normal type to produce is immunoglobulin G, basically. That's the main type of antibody molecule which generally plasma cells produce. Okay, right, so that's how you activate B cells, basically. The B cell initially binds to the epitope of the antigen that it's specific to, okay? It will then internalize it, express, or rather present fragments of that antigen on its surface bound to MHC class 2 molecules, and then wait to receive help by a T helper cell that is directed against that specific fragment of the antigen molecule, okay? and then that will give the B cell the signal to divide and divide and divide. The B cell will then proliferate, producing an entire clone of B cells, which all produce this identical antibody molecule, and then some of those will differentiate then on to become plasma cells, which are the cells which actually start now chucking out a huge number of antibody molecules, and it's estimated to be around 1,000 antibody molecules per second, so it's a huge, huge turnover of antibody molecules. Okay, and they can produce antibody molecules of those different types, basically. So we talked about how the constant region of antibody molecules can be changed, basically, and the main type that you produce is immunoglobulin G, which has functions in activating complement and also in opsonization. Okay, right, so that now concludes our discussion of the adaptive immune response. In the next video, what we'll start talking about is what happens in rheumatoid arthritis. Okay, so we'll start talking about initially the MHC class 2 molecules that give you a huge risk of developing rheumatoid arthritis, which is HLA-DR4.